Hello everyone, and thank you for joining my presentation that investigates neurodegenerative disease-related Cofilin-Actin rod dynamics using a novel optogenetic approach. My name is Davis Roberts, and I am the lead undergraduate researcher on this project under the supervision of Dr. Robert Hughes at East Carolina University. Actin-cofilin rods are commonly found in neurons of patients with many diseases such as Alzheimer's, Parkinson's, and Huntington's disease. These rods normally form under oxidative and energetic stress conditions, ultimately leading to synapse loss in the neurons. In our lab, we have created a light-activated switch named CofActor for monitoring Cofilin-Actin rod formation in living cells. This system can be shown in Figure 1. Using this system, we can monitor real-time Cofilin-Actin interactions in cells undergoing applied energetic or oxidative stress. In previous work, we used the CofActor system over short timescales of around 10 to 20 minutes. Using the data from our previous work, we decided to use the CofActor system over a longer timescale from 3 to 4 hours long. In this experiment, we specifically asked whether the rounded Cofilin-Actin clusters formed over the short time scale could eventually transition to linear Cofilin-Actin rods over an extended time period. Our findings demonstrate that Cofilin-Actin clusters can transition to linear Cofilin-Actin rods, and that they provide a unique way to assess the dynamics of Cofilin-Actin rod formation in living cells. In our previous work that I mentioned before, our CofActor system demonstrated that under stress conditions, actin and cofilin clusters would form and would also combine with other clusters to make larger structures. However, these clusters were very different from distinct native actin-cofilin rods that have a very sharp needle-like structure. These prototype rods were crude, somewhat linear, clumpy structures that were not similar to the native rod structure. Spontaneous rod formation was also very common in our previous experiments. They seem to appear independently of the actin-cofilin clusters. Moving into our current work, Figure 3 shows the rapid formation of actin and cofilin clusters within the first 15 minutes, and the number of clusters is measured in a graph. Figure 3 also demonstrates that actin and cofilin co-localize into clusters under these energetic and oxidative stress conditions. Figure 4 shows the interactions between the clusters after the initial time period of 15 minutes. Following the white arrows, you can see smaller clusters begin to form together to make larger clusters. The most significant finding of this experiment is shown in Figure 5. Here, it is shown that clusters can become the site of a cluster-seeded rod formation event. This cluster-seeded rod appears to emerge from the cluster and elongate over time. As the rod grows in length, the cluster from which it originated shrinks and begins to be incorporated into the rod structure. Spontaneous rod formation, as seen in previous experiments, also occurred within this cell, indicating that both the cluster-seeded rod formation and spontaneous rod formation events can occur within one cell at the same time. In the future, we plan to further study the cluster-seeded rod formation event. With the success of extended time trials, the trials could be extended even further while trying to prevent the cells from collapsing, which is an event that appears to occur in all cells that develop these abnormal actin and cofilin structures. These experiments also need to be done in other cell lines to see if the conditions are constant across cell lines. Eventually, these experiments should be done in neuronal cells to see if conditions are the same.